The essence of cellular respiration is to break the glucose down to make ATP. Here is the equation of cellular respiration. The reactants are glucose and oxygen. The products are carbon dioxide, water, and ATP. Cellular respiration has 4 stages. The first stage is glycolysis. Glycolysis is an ongoing process in all cells. It takes place in the cytoplasm before cellular respiration, and it does not require oxygen. Glycolysis makes a small number of ATP molecules in mitochondria, but its other products are much more important. If oxygen is available, the products of glycolysis are used to produce more ATP molecules through cellular respiration. The process of glycolysis can be summarized as: first, 2 ATP molecules are used to energize a glucose molecule. The glucose molecule is split into 2 3-carbon molecules. A series of enzymes and chemical reactions rearranges the 3-carbon molecules. Second, energized electrons from the 3-carbon molecules are transferred to molecules of NAD+. Molecules of NADH are formed. A series of reactions converts the 3-carbon molecules to pyruvate, which enters cellular respiration. 4 ATP molecules are made. In this process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. The second step is link reaction. Link reaction occurs in the matrix, and it is across the membrane. The stage involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and the carbon is lost, forming carbon dioxide. The third step is citric acid cycle, and it is also called Krebs cycle. It occurs in the matrix. In this process, 1 ATP, 1 FADH2, and 3 NADH is produced. First, acetyl coenzyme A adds its 2-carbon acetyl group to oxaloacetate, producing citrate. Citrate is converted to its isomer, isocitrate, by removal of 1 water molecule and the addition of another. Isocitrate is oxidized, reducing NAD+ to NADH. Then the resulting compound loses a carbon dioxide molecule. Another carbon dioxide is lost, and the resulting compound is oxidized, reducing NAD+ to NADH. The remaining molecule is then attached to coenzyme A by an unstable bond. Coenzyme A is displaced by a phosphate group, which is transferred to GDP, forming GTP, a molecule which functions similar to ATP. GDP can also be used to generate ATP. 2 hydrogens are transferred to FAD, forming FADH2 and oxidizing succinytate. Addition of a water molecule rearranges bonds in the substrate. The substrate is oxidized, reducing NAD+ to NADH and regenerating oxaloacetate. During oxidative phosphorylation, chemiosmosis couples electron transport to ATP synthesis. In ATP synthesis, firstly, hydrogen ions falling down their gradient under a channel in a stator, which is anchored in a membrane. Secondly, hydrogen ions enter binding sites within a rotor, changing the shape of each subunit so that the rotor spins within the membrane. Thirdly, each hydrogen ion makes 1 coupled complete term before leaving the rotor and passing through a second channel in the stator into a mitochondrial matrix. Fourthly, spinning of the rotor causes an internal rod to spin as well. This rod extends like a stalk into the knob below it, which is held stationary by part of the stator. Finally, turning of the rod activates catalytic sites in the knob that produce ATP from ADP. During chemiosmosis, the protons flow back their gradient via ADP synthesis. During chemiosmosis, the protons flow back down their gradient via ATP synthesis, which is built into the membrane nearby. The ATP synthesis harnesses the proton motive force to phosphorylate ADP, forming ATP. Together, electron transport and chemiosmosis make up oxidative phosphorylation. The hydrogen ions combine with the oxygen to form the water. During cellular respiration, 30 or 32 ADP is totally produced. Photosynthesis is the process that converts solar energy into chemical energy. Leaves are the major locations of photosynthesis. The green color is from chlorophyll 2, the green pigment within chloroplasts. The chlorophyll 2 is in the membranes of thylakoids. Light energy absorbed by chlorophyll 2 drives the synthesis of organic molecules in the chloroplast. Carbon dioxide enters and oxygen exits the leaf through microscopic pores called stomata. Photosynthesis can be summarized as the following equation. The reactants are carbon dioxide, water, and ATP, and the products are glucose and oxygen. Chloroplast splits water into hydrogen and oxygen, incorporating the electrons of hydrogen into sugar molecules. Photosynthesis consists two stages, the light reactions and the Calvin cycle. The light reactions are the steps of photosynthesis that convert solar energy to chemical energy. A primary electron acceptor in the reaction accepts an excited electron from chlorophyll 2. Solar power transfer of an electron from chlorophyll 2 molecule to the primary electron acceptor is the first step of the light reactions. The thylakoid membrane is populated by two types of photosystems that cooperate in the light reactions of photosynthesis. Photosystem 2 and Photosystem 1. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons are lost by photosystem 2 are replaced by a process called photosynthesis, which involves the oxidation of a water molecule producing free electrons and oxygen gas, while this electron gas is a byproduct of photosynthesis. It is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthesis, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shedded to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH. When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light reactions are used in the stroma to fuel the Calvin cycle reactions. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce the G3P. The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to RuBP in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, RuBP is regenerated. For every 3 terms of the cycle, G3P are used to reform 3 molecules of RuBP. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Therefore, the Calvin cycle has to run 6 times to produce 1 molecule of glucose.